Hi, I'm Ximeng from Simpson Lab of Nonlinear Optics at Purdue University, and I'm here today to share with you nose microscopy and its application in pharmaceutical analysis and tissue imaging. In pharmaceutical industry, an active ingredient must typically be sufficiently water-soluble to be emitted by blood, but also hydrophobic enough to pass cell membranes. A target compound is normally categorized using biopharmaceutics classification system. Class 1 compounds are the most ideal candidates, while compounds from the other classes may cause problems when used in the production of drugs, such as reduced shelf life, low effective dosage, and these can be costly for the company. Polarization-dependent imaging methods can provide rich local structural information and help facilitate the screening process. The goal of nose microscopy is to evaluate local structure and sample orientation using a set of polarization-dependent second harmonic generation measurement. SHG is a nonlinear optical process that's particularly sensitive to polarization modulation. With our instrument, we're able to acquire up to 30 polarization-dependent images within a second. A set of software were developed to connect intensity to the structure. First, the phase of the modulation was determined and used to convert intensity to a set of polynomial coefficients. Then the coefficients are expressed by a set of Jones tensors in the laboratory frame, which describes the efficiency of SHG process given a particular combination of polarizations. Very often, the local reference frame defined by the sample does not line up with Jones frame. But luckily, with the knowledge of sample orientation, Jones tensors can be converted to local tensors. And last, with the application of crystal symmetry, the total number of unknowns can be reduced from 27 to just a few. Here are some results from naproxen crystal and collagen tissues. A set of 10 intensity images was converted to 5 coefficient images, and a superimposed color map of the coefficient is generated. This analysis can be done at video rate. As shown, the contrast depends both on the local structure of the sample and sample orientation. Typically, knowledge of sample orientation is required to solve for local tensors, and vice versa, but an iterative approach can be used to solve for both. The algorithm starts with an initial guess for the two and iterates between them until the system converges. This approach can be used to recover a single set of local tensors or performed on a per pixel basis. Local tensors reflects the property of local structure of the sample and can be used for sample discrimination. Thank you for listening.